Welcome to the Deep Dive, the show that takes complex cellular machinery and gives you the shortcut to being well-informed.
Today, we are strapping on our hard hats and going deep inside the engine room of life.
We are looking at the structure that makes, well, pretty much all active cellular processes possible.
We're talking about the mitochondrion.
The critical organelle responsible for manufacturing that universal energy currency, ATP.
That's a perfect way to start. I mean, if you think of a cell as this incredibly sophisticated, dynamic factory,
Okay.
the mitochondrion is its centralized power plant. And it operates with just an astonishing degree of efficiency and precise control.
Our source material for today defines it in a very specific way, not just as a powerhouse, but as an energy-transducing compartment.
I love that term.
Me too, because it immediately tells us three things about membranes and how mitochondria just take the game to a whole new level.
Right.
We know cell membranes create separation, which is compartmentation. That lets specific chemical reactions happen without interference. We also know membranes are highly selective, controlling what gets in and out. That's transport.
Yeah.
But energy transduction, that's the real biological marvel we're here to explore today.
It is. It's the third and really the most distinctive role of the mitochondrial inner membrane, a role it only shares with the membranes inside, plastids like chloroplasts.
So what is energy transduction exactly?
It's the conversion of energy from one form to another. Specifically here, we're converting a chemical energy from oxidation-reduction reactions.
So from our food.
From our food, yes. Literally pulling high-energy electrons off those molecules and translating that into a physical force to synthesize ATP.
So it's a chemical reaction being turned into a physical force, which is then used to synthesize another chemical, ATP, which the cell uses for everything else.
Precisely. That ATP then becomes the usable chemical energy that fuels nearly every other energy-requiring process you can think of. Oh, building new proteins in the cytoplasm, the active transport systems on the plasma membrane that maintain the cell's internal environment, you name it.
So our mission for this deep dive is to trace that whole scientific journey. We'll start with how early scientists even spotted this thing and gave it a name.
Then we'll get into its unique double-membraned architecture and how that dictates its function.
With a big focus on how it controls transport, right?
An intense focus. And finally, we'll drill down into the core process, how that flow of electrons generates the force required to power life.
And of course, what happens when that very delicate system breaks down in human disease.
Exactly.
Okay, so to begin, we need to go back in time a bit, to the late 19th century.
Right, when cell biologists, or cytologists as they were called, were meticulously peering into their microscopes, they were still, working to define the very structures inside the cell.
And they had identified these tiny granules inside almost all cells from higher organisms.
They knew these granules were everywhere, just ubiquitous. But their function, completely unknown. They were just structural curiosities at that point.
So who gave them their name?
It took C. Benda, who was observing cells during the very dynamic process of spermatogenesis. He described their often thread-like appearance and gave them their lasting name, mitochondria.
And the name is beautifully descriptive, isn't it?
Yeah.
That comes from the Greek mitos for thread and chondros for granule.
It is.
But how long did it take for scientists to move past just describing their shape and start figuring out what they actually did?
Not too long, thankfully. And the clue came from a surprisingly simple sort of visual trick. In 1900, the famous biochemist L. Michaelis,
The same Michaelis from Michaelis-Menten kinetics.
the very same, he discovered something key. The dye Janus Green B specifically and selectively stained mitochondria in living cells.
And why was that so important? Why was a specific dye such a huge functional clue?
Because of the chemistry of Janus Green B, the dye only becomes colored, visible under the microscope when it is in its oxidized state. Okay, so if the mitochondria were staining the dye.
They had to be actively oxidizing it. Michaelis made the astute leap that if they were oxidizing the dye, they must be the primary cellular oxidizing agents.
So the place where active respiration was happening.
That's it. The staining wasn't just descriptive. It was essentially a real-time chemical assay predicting their role as the cell's burner.
And that was later confirmed.
It was, yeah. Scientists realized the dye's structure actually resembles the coenzyme FAD, and that its oxidation in the cell is carried out by an enzyme called mitochondrial cytochrome oxidase.
And Michaelis also saw that they weren't just static dots.
No, not at all. He noted that these stain structures were highly dynamic. They were moving rapidly in the cytoplasm, changing shape, coalescing, even dividing. It showed they were complex living entities.
So even before powerful biochemical tools, we had a pretty good sense of their function and their physical dynamism.
That's right. And structural conformation also progressed at the same time. As early as 1888, A. Colliger isolated them from insect flight muscle. And what did he find?
He demonstrated that they were osmotically active and were definitely surrounded by a membrane, but the real breakthrough for functional analysis came much later.
When was that?
We couldn't really study their biochemistry until the late 1940s, with the refinement of differential centrifugation.
Right, the technique that lets you spin down cell parts and separate them by size and density.
Precisely. That large-scale isolation finally allowed them to conclusively identify the major pathways that were localized to the mitochondrion.
Which were?
The breakdown of fatty acids, the citric acid cycle, the entire electron transport chain, and of course, ATP synthesis.
So this was the era where the mitochondrion went from being a histological curiosity to the known powerhouse of the cell.
Exactly.
Okay, so by the 1950s, we knew what they did. But it took the electron microscope to really appreciate how they did it.
The source material notes that G. Palade in 1956 provided the fundamental structural definition that we still rely on today.
And that definition is the key to understanding the whole energy conversion process.
It is. The mitochondrion is fundamentally built from two distinct membranes, which in turn define two separate chambers or spaces.
Let's visualize that. You have the relatively smooth outer membrane on the outside.
Right.
And just inside that, you have the intermembrane space, which is this narrow gap, only about 8 to 10 nanometers wide.
And then, crucially, you have the inner membrane.
Which bounds the big central space, the matrix.
Exactly. And the inner membrane is where all the magic happens. Its structure is anything but smooth.
It has these complex, laminated folds that project deep into the matrix. These are the cristae.
Yes, the cristae. And those folds are the key architectural feature that distinguishes the mitochondrion from, say, simpler organelles.
They're not always the same shape, are they?
No, absolutely not. The structure of the cristae can vary. They are often arranged as parallel plates, which is the classic image you see in textbooks.
But sometimes they're different.
But in certain tissues, like the adrenal cortex, or in some protozoans and insects, they can be tubular. This variety suggests some evolutionary adaptation, and maybe that tubular structures are a more primitive form.
But the main point is consistent, right?
Yeah.
Folds equal more surface area.
That brings us to the first major structure-function relationship. It's the ultimate in biological real estate management.
Why do highly metabolically active cells, like muscle tissue or liver cells, have mitochondria just packed with cristae? Sometimes there's barely any room for the matrix.
Because the entire machinery for electron transport and ATP synthesis is embedded in that inner membrane.
So more folds.
Means more membrane, which means more surface area for energy production. A cell that relies heavily on aerobic respiration needs the maximum possible number of these enzymatic machines.
So if you compared a liver cell to a heart muscle cell.
The heart muscle cell, which is constantly beating, will have far, far denser cristae. The density of cristae is a visual indicator of the cell's energy demands.
Generally speaking.
Generally, yes, though you always find these fascinating exceptions.
Like what?
Well, if you study rice that's been germinated anaerobically without oxygen, the mitochondria still develop abundant cristae.
Even though they can't run the full electron transport chain.
Exactly. Even though they lack the necessary enzymes, it shows that the structural development pathway is, somewhat decoupled from the immediate functional need.
Okay, what about size and number? The mitochondrion is usually a sphere or a rod, maybe 1 to 4 micrometers long.
Right.
But the number per cell seems to vary wildly.
It varies tremendously, all depending on metabolic activity. A large frog oocyte might contain up to 300,000 mitochondria, though they only occupy a small part of the cell volume.
And the liver cell.
A single liver hepatocyte might only have about 1,300, yet they take up 20% of the total cell volume. So it's not just about number, it's about density.
And the count itself can be tricky. I remember the sources mentioning giant mitochondria.
This is a perfect example of cellular plasticity. Reconstructions using serial sections, basically slicing the cell up and rebuilding it in 3D, have shown something amazing.
Which is?
What appear to be many small discrete organelles in, say, yeast or rat nephrons might actually be portions of a single giant, highly branched mitochondrion.
So it's one big interconnected network.
It can be. And this structure can then fragment into smaller units when, for example, yeast enter a stationary phase of growth.
That is incredible. So the mitochondria are constantly monitoring the cell's status and fusing or fragmenting based on need.
And their location is often not random at all. They are strategically deployed right where the ATP is needed most.
So in muscle tissue
They're clustered right next to the muscle fibrils, ready to supply energy for contraction.
and in kidney tubules.
In epithelial cells with high transport demands, like kidney tubules, they're situated right next to the extensive plasma membrane folds, directly powering active transport. It's all about minimizing the distance the ATP has to travel.
It's biological efficiency at its best.
It truly is.
Now let's dive into the chemistry of these structures. Once you isolate them, we find that mitochondria are mostly protein and lipid.
With protein making up the majority, yes. But if you look closer at the two membranes, the outer and the inner, you see why the mitochondrion works. Their differences are functional necessities.
And how do biochemists study those differences?
They use a process called subfractionation. They can treat the mitochondria with mild detergents, like digitonin.
And that does what?
It preferentially solubilizes the outer membrane, leaving behind the crucial internal component, the
The
mitoplast.
mitoplast being the inner membrane wrapped around the matrix.
Correct.
Okay, so let's contrast the two, starting with the outer membrane. It's often described as being relatively leaky.
Yes. It's richer in lipid compared to the inner membrane. Its lipids also have more saturated fatty acids, which makes the outer membrane a bit more rigid.
But the crucial functional point is its high permeability.
Exactly. It acts like a molecular sieve, allowing pretty much all molecules up to a molecular weight of about 5,000 to pass freely into the inner membrane space.
Things like sucrose, nucleotides.
All of that.
Yeah.
And this permeability is largely thanks to a single, highly specialized protein.
And that protein is porin.
Correct. Porin forms these stable aqueous channels, roughly 2 nanometers in diameter. Any molecule smaller than a molecular weight of about 6,000 can just sieve right through.
So it's a controlled leak. It makes sure the inner membrane space has the same concentration of small stuff as the cytoplasm.
It does, but porin isn't totally passive. There's evidence it might be voltage-gated and sensitive to anions.
And it also acts as a docking site, right?
It does. We see that certain cytoplasmic enzymes, like hexokinase, which consumes ATP and produces ADP, often bind right at the porin channels.
Why is that significant?
Well, ADP stimulates contact between the inner and outer membranes. So when hexokinase binds to porin, it's generating ADP exactly where it's needed, right near the point of entry. It's an elegant piece of logistics.
Now let's flip to the inner membrane. It's almost the mirror image, chemically and functionally.
It's the wall. It's highly impermeable, functioning much like the cell's own plasma membrane. Only very small, non-polar molecules can cross passively.
And chemically it's different too.
Much different. It's richer in protein, about 80% protein, and it has a high content of cardiolipin, a unique phospholipid that's actually used as a specific marker for the inner mitochondrial membrane.
And it's more fluid.
Much more fluid because its lipids are rich in unsaturated fatty acids. But its extreme impermeability is the foundation of energy transduction.
If the inner membrane were leaky, the entire energy system would just collapse.
Exactly. The integrity of that barrier is the whole game. The only way small polar molecules, ions, or metabolites can cross that inner wall is through specific protein channels or carriers.
Which we call translocases.
And that selective transport is what allows the mitochondrion to carefully control the flow needed for energy production.
So the structural definition of two membranes, two spaces, gives us 4 distinct functional compartments.
Right. The outer membrane, the intermembrane space, the inner membrane, and the matrix. And the matrix holds about 2/3 of all the mitochondrial protein.
And defining where specific enzymes live gives us a kind of biochemical map of the organelle.
It does. Let's start with the outer membrane. Its marker enzyme in mammals is monoamine oxidase, or MAO.
MAO is a huge player in neurological function. It catalyzes the oxidative removal of amino groups from biogenic amines.
Things like epinephrine, norepinephrine, dopamine, serotonin, all critical neurotransmitters and hormones.
So by putting MAO on the outer mitochondrial membrane, the cell has a built-in regulator.
A regulator to control the concentration of these vital signaling molecules that are available for secretion, and this leads directly to its pharmaceutical relevance.
This is where it gets really interesting for you, the listener. Because MAO controls these chemical messengers, drugs that target it are highly potent. MAO inhibitors are used clinically to treat depression.
And the source material also notes that acetaldehyde, a key metabolite of alcohol, inhibits MAO activity specifically for serotonin.
So the buildup of serotonin from that inhibition might contribute to some of the effects of alcohol intoxication.
That's the hypothesis, yes. It illustrates how a single enzyme on the outside of a microscopic organelle can profoundly affect complex processes like mood and neurological signaling.
Okay, moving inward, the inner membrane space acts like a holding area,
It does. It contains enzymes like nucleoside phosphokinases.
and their job is
They're essential because they allow nucleotides to be efficiently phosphorylated and prepared for translocation into the matrix from the cytoplasm.
making sure the organelle has the resources it needs.
Exactly. And finally, we hit the mitoplast, the inner membrane, and the matrix, the real hub of energy
The
metabolism.
matrix is the factory floor.
It hosts most of the citric acid cycle enzymes, which take those input molecules and oxidize them to CO2, generating the reduced coenzymes NADH and FADH2.
And it's also where fats are broken down.
Yes, fatty acid beta oxidation happens there. The inner membrane, then, is the power grid itself, the site of the electron transport chain and the oxidative phosphorylation machinery. The spatial separation is the definition of metabolic efficiency.
So we've established the inner membrane is a highly selective wall. Moving necessary ions and metabolites across it is the next big logistical challenge.
It is. And transport across this barrier has three key requirements that are all tightly interdependent.
Okay, what's the first one?
The first is structural. You have to have specific translocase proteins to ferry the molecules across,
Second, because you're moving things against a gradient sometimes, it's often active transport.
meaning it requires an input of metabolic energy, yes.
And the third requirement is maybe the most fascinating. Any movement of charged ions disrupts the electrical potential across that membrane.
And that disruption is fundamental because the electrical potential is the energy used for ATP synthesis. So ion transport and energy production are intrinsically linked. Move an ion and you directly affect the cell's energy economy.
Let's look at calcium, Cal++, a ubiquitous signaling ion. Our sources highlight that animal mitochondria accumulate vast amounts of it.
They do, especially when they're actively engaged in electron transport. And this accumulation serves two critical purposes.
The first is storage.
Right. When Cal++ accumulates with phosphate, dense granules of calcium phosphate can form in the matrix. These granules are a potential storage pool, maybe used later for processes like bone or eggshell formation.
But the more immediate impact is regulatory, isn't it?
Absolutely. The mitochondrion acts as a major regulator, pulling Kal++ out of the cytoplasm and lowering its concentration.
And since cytoplasmic K++ is a trigger for everything from muscle contraction to glycogen breakdown.
Controlling its level is essential for cellular signaling. A hormone can trigger the mitochondrion to quickly release that stored K++ back out, effectively amplifying the cell's response.
So it's not just a storage tank, it's a crucial buffer and signaling component.
And an interesting counterpoint is that plant mitochondria don't really prioritize K++ psi. Their main ion transport mechanisms focus on phosphate, K plus I, and Mg++ I.
We can also interfere with these processes using experimental tools like ionophores.
Right. Ionophores are hydrophobic antibiotics like valinomycin that can dissolve in the lipid bilayer.
And they can carry ions across.
They can effectively carry monovalent cations like K plus I across the membrane. But this completely bypasses the cell's natural controlled transport systems.
And by carrying charge across the membrane without going through the proper channels, they collapse the ionic gradients.
Exactly. They divert energy away from ATP synthesis. They're powerful tools for proving the existence and necessity of that gradient.
Okay, so the matrix is the metabolic core. Given the inner membrane's impermeability, how does the cell ensure a constant high-volume traffic of raw materials like pyruvate, phosphate, and ADP?
With the specific protein translocases, these are highly specific transport systems.
And they have classic transport traits, right? Substrate specificity, they can be saturated, they can be blocked by inhibitors.
All of the above?
So let's detail the necessary traffic flow. How does the fuel get in?
First, you need the pyruvate carrier to import pyruvate, the end product of glycolysis.
What about fats?
For fats, you have the specialized fatty acid carrier. Fatty acids can't cross as fatty acyl-CoA, so they have to be coupled to a helper molecule, carnitine.
Carnitine carries the fatty acyl group across the inner membrane.
Right. Once inside, the fatty acid is released, carnitine is exported back out, and the fatty acid undergoes beta oxidation.
That's a complicated multi-step transport mechanism just to get one fat molecule across the wall.
It's a necessary complexity. And then you have to import the other ingredients. The phosphate carrier is essential for bringing in inorganic phosphate, one of the two precursors for ATP.
But the truly critical logistics system, the one that makes the whole energy economy run, is the one that exchanges the currency itself, the adenine nucleotide carrier.
This carrier is immensely studied. It ensures that the power produced in the matrix gets exported to the cytoplasm, and the spent currency ADP gets imported back for recharging.
And it catalyzes a strict one-to-one exchange.
A strict 1.1 exchange. 1 ADP in for 1 ATP out.
And what's fascinating here is that this exchange is not electrically neutral.
No, it's electrogenic. And this is a key nuance. ATP carries 3 negative charges, while ADP carries 2. This means for every exchange, a net negative charge moves out of the matrix.
And this directionality is driven by the very electrochemical gradient that the electron transport chain creates.
Exactly. The gradient helps pull the highly charged ATP out and the less charged ADP in. The energy system is basically subsidizing its own logistics.
That's amazing. It's rigged to prefer exporting the finished product.
It is, and the identity of this carrier was confirmed using highly specific toxins.
Right, like atractyloside and boncreic acid.
The thistle toxin atractyloside selectively blocks ADP binding, while the bacterial toxin boncreic acid blocks ATP binding. These are indispensable tools in isolating and characterizing this vital protein.
And beyond the currency itself, we need other carriers to link the matrix to other major cellular pathways, don't we?
Of course, things like the dicarboxylate carboxylate carriers and the glutamate aspartate carriers are essential for integrating the citric acid cycle with cytoplasmic processes, like the urea cycle. Without them, the whole metabolic network of the cell would fracture.
This brings us to a major transport challenge, a logistical crisis known as the NADH dilemma.
Ah, yes.
Glycolysis happens in the cytoplasm and it produces NADH. The cell has to reoxidize that NADH back to NAD+, so glycolysis can keep going.
But the dilemma is simple. The inner mitochondrial membrane is completely and utterly impermeable to cytoplasmic NADH.
So you have all this stored reducing power right next to the power plant, but you can't get the coenzyme itself inside.
You can't, so the cell solves this with an elegant proxy system, or a shuttle.
How does that work?
Instead of sending the NADH molecule itself, the reducing equivalents, the high-energy electrons, are transferred from NADH to a proxy molecule in the cytoplasm.
And that proxy molecule can cross the membrane.
It can. Once inside the matrix, the proxy molecule transfers the reducing power to the mitochondrial NAD+, or FAD, and the newly oxidized proxy molecule shuttles back out to repeat the cycle.
The first example of this is the glycerophosphate shuttle.
Right. This system uses an oxidized proxy dihydroxyacetone phosphate. Cytoplasmic NADH reduces this into alpha-glycerophosphate.
Alpha-glycerophosphate can get across the inner membrane.
It can. Once inside, a membrane-bound enzyme converts it back to dihydroxyacetone phosphate, but in the process, it reduces FAD to FADH2.
So the net result is that the cytoplasmic NADH reducing power has been transferred to FADH2 inside the matrix.
Precisely. FADH2 then enters the electron transport chain. The limitation is that FADH2 yields slightly less ATP than NADH, so you lose a bit of energy efficiency.
But you solve the transport problem.
You solve the problem.
And the second, more intricate system is the malate-aspartate shuttle.
This one is more complex, involving at least 2 separate inner membrane translocases. It's a multi-molecule 4-step cycle that's energetically more favorable.
Why more favorable?
Because the reducing power ends up reducing NAD+ directly in the matrix, so you get the full ATP equivalent. This shuttle is typically active in tissues like the liver, where maximum efficiency is paramount.
Okay, we've built the powerhouse and established its complex logistics. Now we come to the main event. Energy transduction.
Right. The mitochondrion has to take the vast potential chemical energy locked in carbohydrates, lipids, and proteins and convert it into the versatile, usable form of ATP.
And this all converges in the matrix.
It does. Regardless of whether you eat a piece of bread, a snake, or butter, the energy ultimately flows through one central process: the oxidation of reduced compounds to CO2, primarily through the citric acid cycle.
And this oxidation is tightly coupled to the reduction of coenzymes.
Taking NAD and FAD and turning them into NADH and FADH2.
So we can summarize the entire flow of energy generation.
Yeah.
Foodstuffs,
Funnel into the citric acid cycle,
which reduces coenzyme,
which then power the electron transport chain.
which finally generates ATP.
That's the chain of command. Carbohydrates yield pyruvate. Pyruvate becomes acetyl-CoA, and the citric acid cycle completely demolishes it, giving off CO2 and those reduced coenzymes.
Lipids yield fatty acids, which are broken down through beta oxidation in the matrix.
which also generates CO2, acetyl-CoA, and large amounts of NADH and FADH2.
And even proteins get used.
Once their amino groups are stripped off, their carbon skeletons enter the citric acid cycle directly at various points, all leading to the same result: complete oxidation to CO2 and the generation of those high-energy coenzymes concentrated right where they need to be in the matrix.
So those reduced coenzymes represent immense potential chemical energy. They're just desperate to give up their electrons.
Right. We measure this tendency using the standard redox potential, or ED dollars, where electrons flow spontaneously from a negative potential, like NADH at DNP 22.2 volts.
To a highly positive potential, which is oxygen, the final electron acceptor, at plus 0.822 volts.
And when you calculate the total potential change from NADH donating its electrons all the way to oxygen accepting them, the total energy difference is huge, 1.14 volts.
Which translates to a massive amount of free energy, about 52,600 calories per mole.
A huge amount. And if all that energy were released in a single burst, it would be violent, damaging, and completely wasteful.
The waterfall problem.
That's it. The cell has to control the flow. The electron transport chain, or ETC, acts as a carefully controlled chemical ladder embedded entirely in the inner membrane.
It's a series of redox carriers arranged in a precise order of increasingly positive potential, parceling out that energy into smaller, usable portions.
And there are specific points along this ladder where the energy drop is large enough to be successfully captured and harnessed for ATP synthesis.
3 key steps, right?
3 key steps where significant free energy is released. The first is early on. The second happens in the middle, around the cytochrome body to C.1 transition. And the third, largest drop, is right at the end when electrons are transferred to oxygen.
That ordered sequence is critical.
Yeah.
How did scientists confirm the exact invariant sequence of all these carriers?
They used incredibly elegant experimental techniques, starting with spectrophotometry.
Okay, how does that work?
Each carrier has a unique absorption spectrum when it's oxidized versus when it's reduced. By watching the shifts in light absorption, they could measure the redox state of every carrier in real time.
And then they added molecular roadblocks.
The inhibitors were the ultimate proof of order. Take the fish poison rotenone. If you add it, it specifically blocks electron transfer early in the chain.
So everything before the block piles up with electrons and gets reduced.
And everything after the block is starved of electrons and stays oxidized. Then you have antimycin A, an antibiotic, which blocks the middle section.
And finally, a lethal poison like cyanide.
Cyanide blocks the final step, the transfer of electrons to oxygen. By applying these inhibitors sequentially and observing which carriers became reduced, they could map the exact order of the ETC ladder.
You can see this clearly with the oxygen electrode experiment.
You can. If you feed mitochondria pyruvate, oxygen consumption starts.
Huh.
If you add rotenone, it stops. The electron flow is blocked.
But if you then add succinate, another substrate oxygen consumption resumes.
And that elegant result demonstrates that succinate bypasses the rotenone block. It means it enters the ETC at a later point, reducing the later complexes directly.
Confirming that the flow is organized into large, discrete lipid-protein complexes embedded in the membrane.
Exactly.
So let's identify those main complexes.
We focus on the 3 main pumping stations. Complex 1, or NADHQ reductase, is the entry point for NADH. Complex 3, or Q-cytochrome C reductase, takes those electrons and passes them to cytochrome cetors. And complex 4, or cytochrome cetyl oxidase, is the exit point, delivering the electrons to oxygen.
And complex 2.
Complex 2, succinate dehydrogenase, is also present and feeds into complex 3, but it's not part of that primary NADH electron flow pathway.
Okay, we've parceled out the energy release. Now we have to capture that energy and store it in ATP, which is an energetically costly reaction.
It is. And the first verification that this capture is efficient came from the PO ratio,
which measures the ratio of phosphate incorporated into ATP versus the oxygen consumed.
Right. The key observation is that if you don't have enough ADP, respiration slows way down. But when you add ADP, the rate of respiration spikes until all the ADP is converted to ATP.
So by measuring the oxygen consumed during that spike, they could calculate the ratio.
And the results confirm the 3 sites of phosphorylation. Substrates like glutamate, which feed in at the start, yield a PO ratio of about 3. Succinate, entering later, yields 2. Ascorbate, entering near the end, yields 1.
So that proved energy capture was happening at 3 defined checkpoints. But what was the connector between electron flow and chemical synthesis?
That was the central mystery. And in 1961, Peter Mitchell provided the radical answer, which earned him the Nobel Prize, the chemiosmotic hypothesis.
He completely rejected the idea of a chemical intermediate.
Threw it out. Instead, he proposed that electron transport drives the vectorial transport of protons, H+ hydrogen ions, out of the matrix, across the inner membrane, and into the inner membrane space.
So it's like running a massive pump. And that pumping action creates what?
It creates a state of stored energy, the proton motive force. The inner membrane space becomes acidic. It has a much higher H+ concentration, a lower pH, and it becomes electrically positive relative to the matrix.
So you're literally building an electrochemical voltage across the inner membrane.
Exactly. The energy isn't stored chemically. It's stored as a physical, electrical, and concentration gradient.
And the energy stored in that gradient is then used to synthesize ATP.
As the protons flow inward back across the inner membrane, down their concentration gradient through the specialized enzyme, the ATP synthase.
It's such an elegant solution, relying entirely on the structural integrity of that inner membrane wall. And we have concrete evidence this gradient exists, right?
We do. We can measure a clear drop in pH outside the matrix and respiring mitochondria. And the measured value of the total electrochemical voltage is about 0.2 volts.
And that's enough to do the work.
It is. The calculation shows that synthesizing one ATP molecule requires a minimum of 2 protons flowing back across. Actual measurements are closer to 3 or 4 because some energy is used just to power the carrier that exports the finished ATP.
But the most convincing evidence came from the uncouplers we mentioned earlier.
The uncouplers are the smoking gun for Mitchell's theory. Ionophores like FCCP or dinitrophenol, which can carry H+ across the membrane, effectively short-circuit the whole system.
They destroy the gradient.
And when the gradient is destroyed, ATP synthesis instantly stops, even though the electron transport chain keeps running vigorously. This definitive proof cemented the chemiosmotic hypothesis as the central theory of energy transduction.
So the chemiosmotic theory demands that the electron carriers have to be arranged asymmetrically across that inner membrane to pump protons outward. How do scientists map their precise location?
They rely on the impermeability of the inner membrane. They use antibodies or reagents that are too big to cross the barrier. If they add an antibody to intact mitochondria and it binds, that carrier must face the outside the intermembrane space.
And if they want to access the internal matrix-facing side?
They use sonication ultrasound treatment. This causes the inner membrane to pinch off and reform into small inside-out spheres called submitochondrial particles.
So the surface that used to face the matrix now faces the outside.
Exactly. So if an antibody only binds to carriers in these inside-out particles, we know those carriers face the matrix.
And this technique was vital for studying the ATP synthase itself. In electron micrographs, it looks like tiny knobs or lollipops projecting into the matrix.
Right, the F1 particles. The breakthrough came from E. Racker's reconstitution experiments. He found he could chemically remove those F1 knobs from the membrane vesicles.
And what happened then?
The remaining membrane vesicles still performed electron transport, they consumed oxygen, but they could no longer make ATP. The isolated F1 knobs floating freely only showed ATPase activity.
Meaning they hydrolyzed ATP, they destroyed it.
They destroyed it. But the magic happened when he put them back. He reconstituted the F1 particles onto the membrane vesicles, and immediately oxidative phosphorylation was restored.
So that proved F1 is the ATP synthesizing enzyme, and it needs the membrane and the gradient to function.
It did.
So let's detail the structure of this F0-F1 complex. It's basically a nanoscale turbine.
It has 2 primary components. F1 is the projecting knob in the matrix. This is the catalytic engine with 3 active sites on its beta subunits. F0 is the membrane-bound portion.
And F0 is the actual channel that protons flow through.
It is. It contains a critical proteolipid subunit that forms the actual proton translocator. We know this because mutants resistant to the antibiotic oligomycin, which blocks the F0 channel, showed an altered proteolipid sequence.
So how does the physical flow of protons through F0 actually result in a chemical bond forming in F1?
The more accepted proposal is the indirect mechanism, or conformational coupling.
Which treats the proton motive force as a mechanical trigger.
Precisely. The flow of protons through F0 induces a rotational or conformational change in the F0 portion. This change is then transmitted mechanically to the F1 catalytic engine.
And that forces the release of already formed ATP.
Exactly. The energy isn't used to make the ATP chemical bond, but to release the tightly bound finished product from the active site.
We've focused on this highly efficient coupling of the proton gradient to ATP. But biology sometimes uses that gradient for a completely different purpose,
Yeah.
generating heat.
Right, a process called metabolic thermogenesis. This happens in brown fat tissue, which is abundant in newborns, hibernating mammals, and adults exposed to cold.
And their mitochondria have a unique protein.
They do. In their inner membrane, they have a third protein, thermogenin, also known as the uncoupling protein.
And thermogenin is nature's emergency bypass switch.
It's a dedicated, regulated proton channel. It allows protons to flow back into the matrix without passing through the F0, F1 ATP synthase.
So it collapses the electrochemical voltage on purpose.
On purpose. The energy stored in the gradient is dissipated directly as heat, rather than being coupled into ATP. The tissue becomes a furnace, generating heat to maintain core body temperature.
Which is essential for newborns.
Absolutely. And it's tightly controlled. Hormones like norepinephrine trigger an intracellular cascade that results in the release of free fatty acids.
And the fatty acids are the switch.
They're the key switch. Typically, the thermogenin channel is masked, but free fatty acids activate or unmask it, allowing proton flow and heat generation to begin immediately.
Given the incredible precision required to maintain that 0.2-volt difference, it's no surprise that mitochondrial defects lead to severe diseases or mitochondrial myopathies.
These diseases are characterized by abnormal mitochondrial structure and function, especially in high-energy tissues like muscle. Under the microscope, muscle biopsies show these characteristic ragged red fibers.
Which are just aggregates of abnormal mitochondria.
Yes, and electron microscopy reveals the underlying structural weirdness. Bizarre crystals in the matrix, concentric, swirling cristae. The functional defects span the entire organelle.
We see defects in all 4 ETC complexes, in the citric acid cycle, and metabolite uptake.
A critical example is Leber's hereditary neuropathy.
The disease that causes acute blindness, primarily in young men.
It's specifically traced to an alteration in subunit 4 of complex I, the NADHQ oxidoreductase. It shows how a subtle defect at the very top of the ETC ladder can lead to severe specific tissue failure in the highly energy-dependent cells of the optic nerve.
The principles of uncoupling also explain historical medical cases, like
Right.
the patient described by Rolfluft in 1959.
This patient suffered from severely loosely coupled mitochondria. Her respiration was insensitive to oligomycin and showed very high oxygen consumption regardless of ATP availability,
So her mitochondria couldn't efficiently translate the proton gradient into ATP.
which forced her body to maintain an extremely high metabolic rate just to generate enough energy to survive, resulting in debilitating muscle weakness.
And uncoupling also provides a plausible hypothesis for the periodic high fevers seen in the genetic disorder porphyria.
In porphyria, patients accumulate heme breakdown products like protoporphyrin, and experiments have shown that protoporphyrin can act as an uncoupler.
So if this is happening in the body, the accumulated products would dissipate the proton gradient as heat.
Offering a direct molecular explanation for the patient's recurring high fevers.
Finally, the sources remind us that this vulnerability is universal. It even affects agriculture.
Indeed. The case of male sterile maize, the T cytoplasm strain, is striking. It's hypersensitive to a common fungal toxin and the insecticide methanol.
And both agents do what?
Both bind to a small protein on the inner mitochondrial membrane and cause ion leakage. That tiny leak collapses the chemiosmotic gradient, stops ATP synthesis, and leads rapidly to cell death. It just underscores that the absolute integrity of that inner membrane is a cornerstone of life itself.
This has been a true deep dive, tracing the mitochondrion from an early observation of a microscopic granule to defining it as the complex, double-membraned organelle that orchestrates life's energy supply.
We've seen that the entire function hinges on creating these highly specialized compartments where the impermeable inner membrane is the foundation.
And the purpose is centered on moving electrons down a chemical redox ladder. And that chemical energy is then converted with exquisite precision into a physical, high-energy proton gradient, the proton motive force.
And we have to conclude by reinforcing that profound structure-function relationship. Every single fold in the cristae, every specific carrier protein, every fraction of that 0.2-volt electrical difference is fine-tuned to maximize energy production.
And when that precise tuning is altered, whether by a genetic defect like in Leber's neuropathy or by a natural adaptation like thermogenesis, the consequences are immediate and dramatic.
They are.
So what does this intense analysis of the energy factory mean for you, the listener?
Well, consider this provocative thought. Your entire energy supply, The power needed for every cell in your body to fire, to think, to move, to maintain itself depends on the coordinated precision of trillions of tiny molecular turbines, the ATP synthase complexes.
All driven by an electrical potential difference of just zero point 0.2 volts across a microscopic membrane.
Right. So if something as small as a mutation in one subunit of a single complex can cause system-wide failure, what does that tell you about the immense organized complexity of the molecular machines that are currently keeping you running, completely unnoticed right now?
Something profound to mull over until our next deep dive. Thanks for tuning in.